We tend to picture our genetic code as this um, pretty fixed instruction manual,
It's sort of set in stone.
Exactly. But what science keeps showing us is this incredible layer of, well, complexity on top of that sequence, ways our cells actually control which genes are used and when. Today we're diving into one really fascinating part of that, DNA methylation. And specifically, we've got this research paper looking at something called divergent DNA methylation.
Right. And DNA methylation, just as a quick refresher, think of it like chemical sticky notes on your DNA.
Okay.
Chemically is adding a tiny molecule, a metal group, usually to a cytosine base in the DNA. But the crucial point, as you said, it doesn't change the A's, T's, C's, and G's themselves.
Right, the underlying code is the same.
Exactly. Instead, these methyl tags, they act more like volume controls or maybe access controls. They influence how easily a gene can be read by the cell.
Like annotations on a musical score, telling the players how to play a certain part.
That's a great way to put it, yeah. Play this loud, this soft, maybe skip this bit altogether.
So to really get into this, we're focusing today on this research paper about divergent DNA methylation. We don't have the exact title handy, but that's the core concept.
And we'll also pull in some background from the Biochemistry 7th edition textbook where it helps clarify the fundamentals.
Got it. So what's our mission for this deep dive?
Well, first, we really want to unpack what this paper is telling us about divergent methylation. What did they actually find? Why do they think these differences happen? And what might the consequences be?
Sure.
And second, we want to connect that back to the broader picture from biochemistry. Do these findings line up with what the textbook tells us, or are there maybe some surprises, some new angles on gene control?
Basically aiming for a deeper, more nuanced understanding of how this all works.
Precisely.
All right, let's jump right in then. This idea of divergent methylation. What's the paper's main point here?
The central idea is that these methylation patterns, they're not nearly as consistent or predictable as we might have thought.
Yeah.
Even when you look at cells that seem really similar, like the same cell type or maybe closely related organisms, or even the same cells just under slight different conditions, the paper shows the methylation landscape can be surprisingly, well, divergent, different.
Okay, divergent means different, but different how? Like, what specifically are they seeing differences in?
Good question. The paper probably points out a few key things. It could be specific regions in the genome that show more variability, maybe places like promoters, the start sites for genes.
Or enhancers.
Or enhancers, exactly. Those regions that regulate gene activity. They might also be looking at the amount of methylation. Is a region heavily methylated in one case and barely methylated in another?
So the density of the tags.
Right. And also where the methylation is. Is it mostly in those CPG islands we often talk about near genes, which are usually linked to silencing? Or are the differences happening elsewhere in the genome in different contexts? The paper should really get into those specifics.
That definitely sounds more complicated than just an on-off switch.
Yeah.
So why does this divergence matter? What are the potential consequences if these patterns vary so much?
Yeah, this is where it gets really interesting. The paper likely explores the ripple effects. If methylation patterns differ, it almost certainly means gene expression patterns will differ too.
Okay, that makes sense.
And that could have huge implications for development, for instance. How one egg cell becomes all the different cell types in a body. Methylation plays a big role in orchestrating that. Divergence could explain some of the variability we see.
And disease too, I imagine.
Absolutely. We know many diseases, especially cancers, have really messed up methylation patterns, aberrant methylation. So understanding why they become divergent in disease could be critical for figuring out how diseases start and progress.
Right.
The paper might even touch on environmental responses. Could divergent methylation be a way cells sort of adapt or react to changing conditions?
That's a really cool idea. Did the paper mention the techniques they used? How do they actually see these differences?
Oh, they'd have to. A key method is probably bisulfite sequencing.
Okay, I've heard of that.
Yeah, it's pretty standard now. You treat the DNA with a chemical, bisulfite, which changes unmethylated cytosines to uracil, but leaves methylated ones alone.
Wow.
Then you sequence it, and the uracils read as thymines. By comparing that to the original sequence, you can map out exactly which cytosines were methylated. The paper might highlight specific types or, refinements of sequencing that were important for spotting this divergence.
Got it. So bisulfite sequencing lets them get that base pair level map.
Okay,
So
yeah.
we're building this picture of methylation being much more dynamic, more context dependent than maybe the standard view, right?
Exactly, with potentially big consequences for gene control and cell behavior.
Now let's bring in the textbook view. How does this paper's focus on divergence connect with what biochemistry 7th ed tells us about the basic mechanisms?
Well, if the paper links divergent methylation to changes in gene expression, we can definitely connect that to textbook chapters on gene regulation. Chapter 12, for example, goes into detail about how gene expression is controlled, the proteins involved, the DNA sequences.
Right, so if methylation changes gene activity, what's the textbook mechanism for that link?
The textbook probably covers a couple of main ways. One big one is through chromatin structure. Chapter 11 talks about how DNA is packaged.
With histones and nucleosomes.
Exactly. Methylated DNA can attract specific proteins that help condense the chromatin, pack it tighter.
Okay.
And when the DNA is packed tight, it's harder for the transcription machinery, the enzymes that read the gene, to get access. So the gene tends to be silenced.
Makes sense. Less excess, less activity.
Conversely, areas without much methylation might be more open, more accessible, making it easier to express the gene. The paper might even point to specific proteins found binding differently in these divergent regions.
So the methyl tags act like recruitment signals for proteins that open or close the DNA packaging.
That's a good way to think about it. And another angle is transcription factors. Chapter 12 covers those proteins that bind to specific DNA sequences to turn genes on or off. Methylation within a transcription factor's binding site can sometimes physically block it from attaching. Or, interestingly, it might even attract a different set of factors, changing the outcome.
So it can interfere with the normal control switches.
It can definitely modify how those switches work.
What about the enzymes that actually put the methyl groups on or take them off? Does the textbook cover those, and could that explain how divergence arises?
Oh, absolutely. Textbooks cover DNA methyltransferases, or DNMTs, the writers that add methyl groups, and also the demethylases, the erasers.
Right, the writers and erasers.
Understanding the different DNMTs, what sequences they target, how they're regulated, that's key to thinking about how different methylation patterns could be set up or maintained in different cells or conditions.
So maybe the paper suggests that differences in the activity of these write-eraser enzymes are behind the divergence they see.
That's a very strong possibility the paper likely explores. It could be differences in the enzyme genes themselves, or maybe differences in the cell signaling pathways controlling them.
Okay, that connects the dots. Now, thinking about the paper versus the textbook, does this research suggest anything really new or different compared to the standard biochem view?
That's always an interesting question. One possibility is that the paper highlights a much greater degree of plasticity or flexibility in methylation than you might typically get from a textbook.
How so?
Well, textbooks often present these fundamental processes in a more generalized, stable way. This paper might be showing that methylation landscapes are actually quite dynamic, much more responsive to subtle changes or variable between seemingly identical cells.
So while the textbook might emphasize methylation for long-term silencing, say, this paper could be showing it's maybe not always that long-term or stable, that there's more variation.
Precisely. Or the paper might identify, unexpected factors influencing methylation, maybe specific signals or environmental triggers that aren't usually emphasized in introductory texts. It could be uncovering new layers of regulation driving this divergence.
And could it challenge existing models, like the basic rules we learn?
Potentially. For example, we usually learn promoter methylation equals gene silencing.
Right, yeah, generally.
The paper might show cases where that correlation isn't so clear-cut. Maybe a methylated promoter still allows some gene activity, suggesting other factors are overriding the methylation signal. Or maybe they find regions with big methylation changes that, surprisingly, don't seem to affect nearby genes much at all. That would force us to rethink the function of methylation in those spots. These kinds of exceptions to the rule are often where the most exciting new science happens.
For sure, those aha, or maybe huh, moments. So, stepping back, what are the bigger implications here? If methylation is this divergent, what does that mean for biology overall?
The implications are pretty broad, as the paper probably argues. For diseases like cancer, understanding this divergence within a tumor, for example, could explain why some cancer cells respond differently to treatment than others. It's called tumor heterogeneity.
Right. Makes sense.
And for developmental biology, it could help us understand how very precise cell types and tissues are formed, maybe explaining some of the natural variation we see.
What about adaptation or evolution?
Yeah, definitely relevant there, too. If methylation patterns are flexible, as this paper seems to suggest, maybe that's a key way organisms can adjust to their environment epigenetically. It touches on epigenetic inheritance, possibly, and even how species might diversify over evolutionary time.
Wow.
And the paper almost certainly points to future research. What are the next questions? We need to nail down the exact mechanisms causing divergence, figure out the precise functional consequences in different systems. Lots more to explore.
Absolutely. Lots of exciting avenues opening up. So to wrap things up, what are the main takeaways from this deep dive into divergent DNA methylation?
I think the core message is that DNA methylation, this really key epigenetic mark, is surprisingly variable. It's divergent, even where we might expect uniformity.
Mhm.
And this variability likely has real impacts on gene expression, development, disease, and maybe even adaptation and evolution. The research connects this to fundamental biochemistry, chromatin, transcription factors, but potentially adds this layer of dynamism and complexity that maybe wasn't fully appreciated.
And for you listening in, especially if you're studying biology, this really highlights just how intricate and dynamic our genomes are regulated. It's not just the DNA sequence itself.
Not at all.
It's these added layers like methylation, providing this sophisticated control that contributes to the complexity and adaptability of life.
Couldn't have said it better. It really adds a crucial dimension to understanding how cells work and respond.
So here's something to chew on as you continue your studies. What might be the actual evolutionary advantages of having such a flexible, maybe even noisy system of gene regulation through methylation? How could that variability itself be beneficial? Definitely some interesting food for thought there.